Good afternoon, everyone. My name is Genesis Blanco Flor Paharliaga. This day, we are talking about the following methods of identification. Number 1, anthropometry. Number 2, portrait parley. Number 3, forensic odontology. Number 4, forensic anthropology. Number 5, forensic serology. And number 6, DNA fingerprinting. Anthropometry, designed by Alphonse Bertillon, began in 1890 and lasted approximately 20 years before being replaced by fingerprint identification. Alphonse Bertillon proposed a method of identification in 1879 using anthropological body measurement with the help of his father. Alphonse Bertillon was successfully developed and implement anthropometry in 1882. In addition to physical measurement, Alphonse recorded physical descriptors of the person, for example, hair and eye color, and including any unusual markings on the body like tattoo and scars. This was known as the portrait parley or speaking portrait that described many personal characteristics. Bertillon was promoted to head a newly formed Department of Judicial Identity for the Paris Police. Due to its success, anthropometry or Bertillonage spread throughout the world, with the United States adopting it in 1887 and Great Britain in 1894. Portrait parley. These two French word means speaking likeness. Portrait parley can be defined as a rigorous system for verbal description of characteristics of the subject. It is based on the Bertillon method of criminal identification by measurement of human body and still in practice today to some extent. In order to recognize individuals who were repeatedly arrested, Bertillon developed means by which portraits could be sorted by common morphological characteristics, the specific shape of the different parts of the face, and those an individual prior photo could be without having to resort to browsing through large collection of portraits. In 1882, Alphonse Bertillon was also called father of scientific detection, said that anthropology could be invoked to aid in the sight recognition and identification of criminals. Employing these methods of scientific description, he worked out a system of identification utilizing 11 skeletal measurements which are practically unchanged after maturity and are not affected by increase or loss of weight. Bertillon stated from the observation that human bone structure was more or less absolutely fixed by the age of 20, and that the skeleton varied tremendously in its dimension between one person and another. Before DNA evidences and before that, the ubiquitous adoption of fingerprinting became the dominant mode in which criminals were identified by law enforcement. Another form of biometric identification was widespread around the world. Forensic odontology is a proper handling, examination, and evaluation of dental evidence, which will be presented in the interest of justice. The evidence that may be derived from the teeth, the age, and the identification of the person to whom the teeth may belong. Kaiser Nelson defined forensic dentistry as the branch of forensic odontology that in the interest of justice deals with the proper handling and examination of dental evidence and the proper evaluation and presentation of dental findings. Odontological identification is based on systematic comparison of pre- and post-mortem dental characteristics of individual based on dental record and the supporting radiographs. But this technique is complicated by the trauma to jaw and inadequate antemortem dental information. According to American Board of Forensic Odontology, dental identification can be divided into 4 types. Number 1 is positive identification. The antemortem and postmortem data match to establish that it is from same identity. Number 2 is possible identification. The antemortem and postmortem data have few consistent features, but because of quality of the records, it is difficult to establish the identity. Number 3, insufficient evidence. The data is not enough to form the conclusion. Number 4 is exclusion. The antemortem and postmortem data clearly inconsistent. In many cases, dental identification, fingerprint, and DNA are most commonly used and often complement each other. Forensic anthropology is the study of human remains that involves applying skeletal analysis and techniques in archaeology to solving criminal cases and for the purposes of establishing identity. A forensic anthropologist can read the evidence in skeleton. They can determine any primary identification such as skeletal or developmental age, biological age, living stature, and ancestry or racial affiliation. When human remains or a suspected burial are found, forensic anthropologists are called upon to gather information from the bones and their recovery context to determine who died, how they died, and how long ago they died. Their services have been of particular value in 3 main areas. Number 1, investigation resulting from homicide, unexplained natural death, and mass fatalities. Number 2, non-criminal events resulting in multiple deaths arising from natural catastrophes. Number 3, war crimes, investigation, and genocide. The anthropologist may also responsible to advise on the manner of death, particularly in cases of skeletal trauma or physical process such as burning, dismemberment, or explosive damage have disrupted the remains, rendering quantification and identification problematical. Of the blood or blood stain is confirmed, further analysis is done to establish other characteristics. The blood stain is initially subjected to blood typing. DNA fingerprinting, also called DNA typing, DNA profiling, genetic fingerprinting, genotyping, or identity testing in genetics. Methods of isolating and identifying variables, elements within the base pairs sequence of DNA. The technique was developed in 1984 by American geneticist Alec Jeffreys after he noticed the certain sequences of highly variable DNA known as mini satellites which do not contribute to the function of genes are repeated within genes. Jeffreys recognized that each individual has a unique pattern of mini satellites, the only exception being multiple individuals from a single zygote such as identical twins. The procedure for the creating a DNA fingerprinting consists of first obtaining a sample of cells such as skin, hair, or blood cells which contain DNA. The DNA is extracted from the cells and purified. In Jeffrey's original approach, which was based on restriction fragment length polymorphism RFLP, was then cut at specific points along the strands with proteins known as restriction enzymes. The enzymes produce fragments of varying length that were sorted by placing them on a gel and then subjecting the gel to an electric current electrophoresis. The shorter the fragment, the more quickly it moved towards the positive pole. The shorter double-stranded DNA fragments were subjected to a blotting technique in which they were split into single strand and transferred to a nylon sheet. The fragment underwent autoradiography in which they were exposed to DNA probes pieces of synthetic DNA that were made radioactive and that bound to the mini satellites. A piece of X-ray film was then exposed to the fragments and a dark mark was produced of any point where a radioactive probe have become attached. The result pattern of mark could then be analyzed. So that is for today. I hope you all enjoy to my presentation. Thank you, goodbye, and peace.